Hello, and welcome to part one of adaptive immune response, also known as acquired immunity. So just a real quick overview of what happens in the adaptive immune response. First of all, we have to be exposed to an antigen to begin with. So we're exposed with the primary response. This is the very first exposure, and the adaptive immune response will take probably several days to respond. Some of that will be antigen specific, so depending on what antigen or pathogen has been introduced in the body will play a role in the length of time it takes for the adaptive immune system to, one, be activated, and then two, move into its differentiation and effector function. So in primary response or first exposure, we first have antigen presentation. This is carried out primarily by dendritic cells, but will also be presented through macrophages and B cells. Once antigen presentation has occurred, then B cells will be activated, T cells can be activated, and of course natural killer cells can also play roles in the adaptive immune response. B cells will differentiate into plasma cells to produce antibodies, and if they've been activated by a T-dependent antigen, then we will also see the formation of memory cells. This immune response is known as humoral immunity. Now, T cells will get activated and differentiate into what are known as CD8 T lymphocytes or cytotoxic T cells, or they are CD4 T lymphocytes known as T helper cells. T helper cells will also subdivide into multiple classes of T helpers. We will look primarily at T helper 1 and T helper 2. The T cell response is known as the cell-mediated response. Now, in the adaptive immune system, again, we have humoral immunity, including B cells, and cell-mediated immunity, including the different classes of T cells. Both come from lymphoid progenitor cells. Now, B cells will begin as a naive B cell when they are in the lymph node. Prior to that, they develop in the bone marrow. Both both B cells and T cells both begin in the bone marrow. And from the bone marrow, where they are what are considered immature, when they reach maturity, they will leave the bone marrow, and B cells will go to the lymph nodes directly. Some may go to the spleen. And T cells will make a stop. So the T cells will go to the thymus, and then they will go to the lymph nodes. So they make a little pit stop at the thymus, which is why they are called T cells. Now, both B cells and T cells, when they leave the bone marrow, will have what are called receptors. Now, B cell receptors are called BCRs. They are an IgM, a class of antibody. They are an IgM antibody in its monomer form, bound to the surface of B cells. T cells have what's called a T cell receptor. Their T cell receptor has a co-receptor on the side of it called a CD marker. CD marker stands for cluster of differentiation. It's part of the recognition process later inside the lymph nodes during antigen presentation. The cluster of differentiation, or the CD4 or CD8 marker, will play a role in antigen presentation. But it also is part of what differentiates our T cells. Now, in its immature state here, we just simply have a CD marker. These B cell receptors are really important. B cells will, once they move into the lymph node, they will become what are called naive B cells. This means that these antibodies that are bound to the surface, again, IgM antibodies, very specific class, that are bound to the surface of these B cells are already specific for certain antigens. All antibodies are specific. So by the time the B cell gets to the lymph node, it has yet to be activated, produce any antibodies to really do anything. It's just in a naive state, sitting in the lymph node, waiting for its specific antigen to show up. T cells are also found in the lymph nodes, also in a naive state. Naive just simply means that the cell is differentiated and knows what it's supposed to react against. It just has not been activated to carry out that reaction. When a B cell is finally activated, it will become either a plasma cell, which is a strict antibody producing cell, fairly short-lived, or it will become a long-term memory cell. The memory cell will already be differentiated, and the memory cells will have a different type of antibody on their surface. Memory cell and plasma cell formation is carried out by a process known as clonal proliferation. We will go into details of each of these. We're just giving an overview of what's occurring here. Now remember, this all here is in the bone marrow. This diagram here that we're looking at. Now T cells are part of the cell-mediated response, and we have two basic classes of T cells that we're going to worry about in this class, which are the T helpers, including T helper 1 and T helper 2s. And we're going to look at cytotoxic T cells, or TCs. We'll talk about T regulatory cells, but not in great detail. And then the last cell of the cell-mediated response are the natural killer cells. And natural killer cells have a very distinctive responsibility. So cell proliferation and adaptive immunity. Here's another type of diagram, just kind of showing what happens in an adaptive immune response. In the innate system, a dendritic cell is going to phagocytize and recognize an antigen. This will trigger one of its toll-like receptors, nod-like receptors, or rig-like receptors. When it enters into the lymphatic system to present this antigen that it has found, along with the co-stimulatory molecules it's producing because its pattern recognition receptor has been bound, it's going to react with naive helper cell T cells and naive cytotoxic T cells. Remember, these have a cluster of differentiation 8, a CD8, and these are CD4s. Now, this dendritic cell is going to be presenting this antigen, and it will either bind to helper T cells or naive cytotoxic T cells. Either way, if it bumps into a helper cell here, it activates the T helpers. Different classes of T helpers are going to then activate B cells. Cytotoxic T cells do not activate B cells. They immediately begin proliferating and moving out into circulation to get to wherever a virus or intracellular infected cell is. Cytotoxic T cells deliver apoptosis messages. They go out and they tell our cells that are infected, usually by viruses, to undergo apoptosis. Naive helper cells, they run around and tell everybody else what to do. They're going to deliver their cytokines. They're going to deliver those signaling molecules. They're going to tell macrophages to become super macrophages. And some of them are going to come over here and tell naive B cells that, yep, that antigen is bad. You need to make plasma cells to produce antibodies, and you need to make a couple of memory cells so that if we ever run across this thing again, we can attack it very quickly. So just a quick review on the overview of the immune system. You definitely want to be able to contrast humoral and cell-mediated immunity. You want to know the difference between primary and secondary immune responses, as well as the CD4 and CD8 receptors on T cells. What is a B cell receptor? Differences between cytotoxic and T helper cells. The three classes of T cells that we're going to study in this particular unit. What's the function of a plasma cell? Which cells of the adaptive immune response are going to form memory cells? And what is the role of natural killer cells? We didn't review this in this lecture, so that would be a good one to maybe look up in the text or to do a little Google search on. So that's it for our overview of adaptive immunity, part 1, and we will move on to the next part, part 2. See you then.